For the water pH and buffers lab, we have quite a lot of supplies that we need to bring to the lab. Over here on this side, I've got the supplies that are provided by the hands-on lab kit. So we're going to need our wells. This is where we're going to be putting liquids that we'll be testing to see if they're acids, bases, or if the pH is more neutral. I've got my 2 plastic cups, and inside of the cups, I put my little short pipettes. And the reason why I did this is I know that these pipettes are going to go all over the place. I'm going to be using these cups in the last experiment, so this will help keep me a little bit more organized today. I've got my test tube brush for washing up later and cleaning the wells. I've got my scale and my little weigh boat. My beaker, my graduated cylinder. It's all sitting on the filter paper that the kit provided. And for our experiments today, we're going to want that great big square sheet of filter paper that the hands-on lab kit provided for us. The other thing that you're going to want to get is the packaged water pH and buffer supply. This has our food coloring and a couple other things that we're going to be using today in our experiments. The reason why they put it into this separate container is because we've got the hydrochloric acid and the sodium hydroxide, and both of these can be a bit more damaging, so we need to handle them with more care. I like the fact that they separated them out so that we can keep them separate and treat them a little bit differently today. The other thing that we're going to need is our kit provided pH paper. We will be making our own pH paper as well today, and the nice thing is we'll get to compare our homemade pH paper, how effectively it works, to how effectively the hands-on lab made pH paper will be. Now, some of the things that we need to bring will be some of the things that we want to test today, especially to see what the pH is. So I went wandering around through my house, and in my house I have a fish tank. So I got some fish water because I want to see what's the pH, especially since it's so important for fish to have an appropriate pH for their health and for their long-term survival. There's also a pond in my neighborhood, so I went and I grabbed some of that pond water as well. And I've got rain barrels at my house. What I'm doing is I'm capturing the water that falls on the roof. I capture it into a big barrel, and then I use that water to water my plants. I wanted to know what the pH of that would be, especially after the water is moved across my roof and potentially picked up some of those molecules from the roof. I've got some oat milk, some tea, contact solution, Bactine, which I use whenever I have a cut. I've got some cleaning solution, some Windex, and then of course some mouthwash, some Listerine. So these are all of the items that I want to know. What's the pH? Do I have an acid, a base, or is it more close to neutral? This will help me to understand, especially with things like my cleaning products, how well those cleaning products are working, and more importantly, why those cleaning products work to do things like kill bacterial cells. Remember, both acids and bases have that ability to destroy the plasma membrane, destroy the cell membrane, and essentially kill our cell. So it'd be nice to know how these products, what their pH is. It'll help me to understand how those products are working to disinfect and to kill bacterial cells. Hopefully with the contact solution, hopefully this shows up as neutral. We want our contact solution to mimic tears. We need that kind of neutral solution. The last thing that I want is to put in contacts and to feel that burn because maybe the pH of my contact solution was not correct. So it'll be interesting to test this today and see if it really does show up as neutral, as I anticipate that it will. A couple other things that we're going to need. We're going to need some cabbage. We're going to be heating up this cabbage. Now, I don't know if you're like me, but the smell of warm cabbage I find kind of disgusting. So I encourage you to either have the windows open if it's a bit breezy outside, or have a fan that's on a low setting if you don't like the smell of warm cabbage. The other thing that we're going to need is baking soda. So we've got our list of supplies here that we are bringing to today's lab. The piece of advice today is to work sequentially, especially for the first part of the lab. In the first part of the lab, you're going to be dealing with surface tension of water and playing around with that. So I guess my final tip for you today is make sure you're not too caffeinated. You want a real steady hand for the first part of the lab, and be prepared to allow some of your experiments to sit for a while. So this isn't a lab that you're necessarily going to do start to finish all at one time. We've got a couple of things in this lab that are going to need to sit for several hours. So you're going to start them, leave and go do something else, and then come back and finish your lab. Now, in experiment 1, once you're done dealing with surface tension and you've recorded your responses, the last part is we're going to get our drinking glass and fill it with a half cup of water. Now, it's not as important here that half cup is exactly perfect. In some experiments later it will be exactly perfect, but it's not for this one. We're going to take our food coloring, and our food coloring comes from our little package of prepackaged materials. And I double checked on our supply list. This is the 1 milliliter amount, so I don't have to do a secondary measurement. So it's kind of nice, they've already got it all measured in for me. You can see when I cut off the tip that a couple of drops of food coloring did escape, and that's okay. And when I pour that food coloring in, what we are going to notice is that food coloring immediately starts to disperse. Our food coloring molecules are demonstrating to us simple diffusion. They're moving from an area of high concentration to low concentration using no energy. Now I'm going to give this water a little bit of a swirl to make sure that those waters reach equilibrium faster. In other words, my motion in helping wiggling and jiggling of those molecules is going to help simple diffusion to happen faster. I've got my piece of celery, and I went ahead and pulled this piece of celery right out of the heart of my clump of celery that I purchased from the store. The end has been cut into an angle, and I'm just going to set it there into my cup of food coloring. Most of the time, the celery that we have in the store has that kind of light color, and the way how that color is achieved is the growers will go and wrap the celery so that it is deprived of sunlight. It makes our celery a lot lighter in color. I've also been growing celery in my garden, and this is the color of what celery should look like when it is immature and starting to grow in the garden. So I'm actually going to test both of these today because I want to see if the store-bought celery behaves a little bit different than my garden celery. And the other thing that you need to know is that in our celery leaf, this is all one leaf, we have little tiny tubes. Those little tiny tubes are made up of xylem cells, X-Y-L-E-M. I talked about them a little bit in my Bio 103 lecture when I talked about the properties of water. And so those cells will stack one on top of each other, and they make essentially a straw going from the roots all the way up to the tippy tips of the leaves. And so what I know will happen is that water will move through my celery plant. Our food coloring should help us to be able to track that movement of water through our celery. Now, in experiment 2, one of the things that you're going to be doing is something that I was really excited about when I was looking through the hands-on lab kit and what they offered for us to do. And what we're going to be doing is we're going to be making our own pH paper. And the way that we're going to be doing this is by using purple cabbage. Purple cabbage has something called anthocyanins. And anthocyanins are a pigment molecule that makes the cabbage look purple. And that pigment molecule can be used as a natural dye. In my Plants and Society class, Bio 113, we actually use and cook purple cabbage to release those anthocyanins, and then we put a bunch of wool in there and we dye the wool to get this kind of beautiful purpley blue color. So we're going to be using the anthocyanins to turn our filter paper into pH paper. The anthocyanins in our cabbage are going to be a natural indicator of pH. If a substance is more acidic, then the anthocyanins will look red. If a substance is more alkaline or more of a base, then the anthocyanins look blue. So when you look at plant products and you see anything that has this kind of purpley blue color, it's pretty safe to assume that inside of the plant cells are anthocyanins. So with the hot water, we release the anthocyanins. Now you can cut this cabbage up into tiny, tiny pieces. And the tinier the pieces, the more surface area is exposed, so the more anthocyanins will get into the water. The reason why that's important is that we're going to take our filter paper. Now we've cut into the 4 equal squares. And the easy trick to that is to fold your paper into quarters, and then you've got your 4 equal squares. So you're going to take one piece of paper, and you're going to completely submerge it in the water. Once you have it completely submerged, you need it to sit in there for 5 minutes. Once it is sat in here for 5 minutes and absorb those anthocyanin pigment molecules, then you set it off on a plate to start to dry. We're then going to use this in a later experiment to test the pH of different substances. So it's kind of cool that in this experiment, we get to learn a little bit more about how to make our own pH paper, and I was happy to bring to you the way to naturally dye fibers by using those anthocyanins. Now for one of our last experiments, we're going to need our well unit, and what we're going to be doing is filling each of these little wells with about half full with some kind of solution. And one of the things I wanted to point out to you is if you look really closely, and for me it helps to have sort of a dark surface underneath, you can see that along the top we have numbers that number each of our columns. So this is column 1, 2, 3, 4, 5, and 6. And then right along this side, what you'll see is that our rows are labeled. So this is row A, B, C, and D. Now, I have a tendency to forget what I put where. And I knew that even though some of my solutions I'm going to be testing have different colors, I would lose track of what I put into each well. And we need to make sure that we know what we put into each of the wells so that we can get an accurate pH. And so what I created was a little table. And I wrote down each of the little wells, and then I wrote what I was going to put in it. So in 1A, I'm putting hydrochloric acid. In 2A, I'm putting the NaOH. And in 3A, I'm putting DI, and DI stands for distilled water. 4A has my fish tank water, 5A my pond water, and etc. This will help me to keep straight what I'm putting in each of my wells. I have been cutting my pH strips in half, and the reason why we're doing this is so that we can use less materials, essentially. Now, whatever you do, do not throw this away, especially the color scale on the top. We're going to need that for determining what the pH of our solution is. So once you have your wells filled, a couple of tips of advice as you are filling your wells. Make sure to use a separate pipette for each solution. We don't want any cross-contamination because that will affect our pH measurements. The other thing to keep in mind, if you haven't used one of these little pipettes before, is that when you squeeze the bulb, you push air out. So we first squeeze the bulb, push all the air out, then stick it into our solution. When we release, it will suck everything up. And my suggestion is to make sure to get some of the liquid in the bulb part up here, the part that you're pressing with your fingers. That way you can accurately fill to about halfway. Now we don't have to get super picky about halfway, that everything is exactly perfect as far as halfway is concerned. We just want to make sure that we have controlled in each of our wells and made sure that we have roughly the same amount of solution in each one. We don't want the amount of solution to potentially impact our experiment. So what we're going to do is we're going to take a little bit of pH tape and we're going to dip it into the liquid. And what you'll notice is immediately it will start to change color. So don't leave it in there very long. If you leave it in there too long, the chemicals on the pH tape will start to, they'll start to leave. And we need those colors to stay there, because once we have our pH tape colored by the water, we then hold it up to our scale to determine the pH of our solution. So I'm testing my fish tank water here, and what it is showing to me right now is my fish tank water is undoubtedly a pH of 8. It has that very deep green that I'm comparing to and seeing on the little strip, the little color chart here. So that means that my fish tank is definitely a base. Now, one suggestion that I have for you when we're dealing with color change like this is to make sure to always do everything against a white background. Sometimes our background color will affect what our pH tape looks like. It will affect what color we perceive it to be when comparing it to the scale. So test all of your solutions. One thing that I do want to point out, I've got tea over here, and tea is already very dark. So when I test my pH paper, I need to keep in mind that because my tea is a darker color, that might be affecting what I'm seeing on the pH tape. So keep that in mind. When you measure your different solutions, the color of the solution may be affecting the color of your pH tape. So keep that in mind. Once you have tested using these commercial pH papers, you've gotten experience with how this is supposed to work. Everything has been controlled for as far as the creation of these pH papers. We know that they're giving us a fairly accurate reading. You're next going to use your homemade pH paper that was made by soaking the filter paper in the cabbage water, grabbing those anthocyanins. Remember, our anthocyanins are a pH indicator. The color change of your homemade pH strips will be compared to the hands-on lab color chart that they have. As the color of the pH tape becomes more pink, we have more of an acidic solution. As that color of your homemade pH tape becomes more of a blue and even into a green, when you look at the hands-on lab little color bar there, that's going to tell us that we have a more alkaline or base solution. I hope you enjoy this lab and have a lot of fun testing things. Also getting really familiar with the different pieces of equipment. In this set of experiments, in this experiment with our wells, our independent variables, the different solutions. Our dependent variable, the data that we're collecting, is a pH number, whether that's 8 or 2 or 1. And we are going to try to control for everything to make sure that our pH number is due to our solution inside of our wells and not due to anything else. If you have any questions, please drop me an email, send me screenshots, send me short videos. I hope you enjoy this lab.